Namaste students, Kushra Vidya, I, JV and Dr. Y. Chandrakala, Assistant Professor Jyoti Vidyapeeth, Women's University, Jaipur. In today's session, we are going to discuss about the intercellular compartment and transport. Intercellular transport uh, mainly take place through protein shorting and vascular transport. That means how a protein gets to their appropriate destination within the cell. And through vascular transport, how a vascular shuttles the protein and the membrane between the cellular compartments. So first we will discuss about the protein shorting. So the primary function of a compartment within a cell. Okay. Let's see what are the primary functions of the cell organelle. The cytosol contain many metabolic pathways, okay, like protein synthesis. Nucleus contain many genomes, and DNA and RNA synthesis take place. Endoplasmic reticulum, the synthesis of the most lipid, synthesis of the protein, and distribution of many organelles, and to the plasma membrane. The Golgi body apparatus modify and sorting and packing of the protein and lipids for either secretion or delivery to the other organs. Lysosomes are the intercellular degradation take place. Endosomes help in the sorting of the endocytosed material. Mitochondria help in the ATP synthesis and by oxidative phosphorylation. Cytoplasm help in the ATP synthesis and the carbon fixation by the photosynthesis. Peroxisome is the oxidation of the toxic molecules. Okay. So, next the organelles important for the protein by three distinct mechanisms. Okay. First is the transport from cytoplasm into the nucleus through a nuclear pores. Okay. Next is the transportation of from the cytoplasm to the organelles by protein translocator in any membrane. Third is the transport from endoplasmic reticulum to the other organelles through a vesicle. Okay. So the signal sequence target protein to their destination. So the function of a signal like import into a endoplasmic reticulum, that is then retention of the lumen in the reticulum, then import into a mitochondria, import into a nucleus, and import into a peroxisome. So positively charged amino acids are like amino acid, and here they like and negative charged amino acid which extend the endoplasmic reticulum retain the signals of the commonly referred signal layer of the amino acids and work accordingly. So the signal sequence are necessary for the sufficient for the protein targeting as per the signals the protein molecules attached to the cytosol to the attached to cytosolic proteins. Okay. So the first mechanism, the protein enters the nucleus via a nuclear pore. So the nuclear membrane is a double membrane or a continuous where the endoplasmic reticulum, both compartments share the same lumen, so perforated by a nuclear pore. Then the nuclear pore complex is a selective molecular gate. So here it composed of more or less 100 different proteins. Small water molecules, soluble molecules, passes freely, and the micromolecules must carry appropriate signals. Here, the GTP hydrolyzes and provides the energy that derives the nuclear transport. So, the nuclear envelope disassembles and reforms during each cell division. That is the interphase nucleus, prophase nucleus, the early telophase, or the late telophase. In the mechanism 2, the protein translocates from the cytoplasm to the organelles, where the protein moving from the cytosol into the endoplasmic reticulum, the mitochondria, cytoplasm, chloroplast, and the peroxisome. So the movement of the protein is mediated by a special protein termed as protein translocators. Unlike the passage through the nuclear pores, the translocation requires unfolding or co-relay translation transport. So the protein are unfolded during the translocation into the mitochondria. The endoplasmic reticulum is a network of membrane sheath and tubules. The active ribosome may be in the cytosol or associated with the endoplasmic reticulum. The ribosomes are directed to the endoplasmic reticulum by SRP and or ER signals. The soluble proteins cross the endoplasmic reticulum membrane into the lumen. So the integrated membrane of a protein into an endoplasmic membrane. So the multiple passes of a protein using internal starts the transfer of a sequence. The third mechanism starts here with a vascular transfer. The integration of a transmembrane protein into a endoplasmic reticulum membrane. So here the multiple paths of the protein use the internal start of the transport sequence. Okay, the next mechanism third where the vascular transport or the transport vesicles continuously burn off these from and fuses to the other membrane compartment, producing a constant flux of a material carry the soluble proteins and the lipids. The membrane proteins in the layer between the compartments are transported along with the microtubules. So here the Catherine coat of vesicles transports electron cargo molecules, and the complex of the Catherine forms the baskets around the vesicles and helps them to pinch from the membrane. Here the movement of the transmembrane to the receptor to the cytoplasmic domain of the receptor binds to the adapting. So the additional molecules, calcium molecules bind to the increasing curvature of the dynamic assembly of a ring forming a Catherine coat bed. So the Catherine coat vesicle transported selected to the cargo molecule. The snare proteins are important for the membrane transfusion via snares, V snares, and T snares. Tightly bind the complex brings the two membrane together to promote the complex binds to the two membrane together to promote the fusion. This session is powered by 2.0 Jyoti Vidyapeeth Women's University, Jaipur. If you have any queries, we'll discuss